update on SPARC strategy and portfolio, hosted by SPARC. As a reminder, all participant lines will be in the listen-only mode, and there will be an opportunity for you to ask questions after the presentation concludes. Should you need assistance during the conference call, please signal an operator by pressing star, then 0 on your touchtone phone. Please note that this conference is being recorded. I now hand the conference over to Mr. Jaideep Israni. Thank you, and over to you, sir.
Thank you, Faisal. Good evening, ladies and gentlemen. I am Jaideep Israni. On behalf of SPARC, I thank you for joining today with us for SPARC's update on strategy and portfolio. On our call today will be CEO Mr. Anil Raghavan and members of SPARC senior management team. The presentation that we will be using for today's discussion was sent out earlier. We hope you have received the slides. The slides are also available on our website, that is www.sparc.life. We will also upload the call transcript on SPARC's website soon. The format of the discussion will be similar to what we have followed in the past. That is, we will go through the presentation first and then open the call for questions. Before we start, I would like to remind you that our discussion today include forward-looking statements that are subject to risks and uncertainties associated with our business that may cause actual results to differ from those projected in the presentation. I will now hand it over to Mr. Anil Raghavan for his presentation.
Thank you, Jaideep, for the introduction. Hello, everybody. A very warm welcome to all of you to the 11th edition of SPARC's annual portfolio review. Thank you so much for joining us today and for your continued support and engagement. Always means a lot to us. We are delighted to welcome you back to this very important call. As usual, as Jaideep said, we have SPARC's management team on the call today with me. In the interest of time, we don't plan to do introductions. I request the presenters to briefly introduce themselves before beginning of their segments. By way of agenda for today, we will start with brief comments on our strategy and certain updates on important programs pertaining to late-stage clinical assets and commercializations. Subsequently, we will dedicate most of our time in the rest of the call to provide more detailed updates on 5 important programs, which are 4 NCE clinical programs plus 1 important biologics platform with clinical visibility. Dr. Su-Long Yao and Dr. Nittan Baumler will provide those updates. We will conclude the presentation with brief comments on our cash flow situation and financial performance from Chetan. That's Chetan Rajpara, our Chief Financial Officer. So with that, let's get started with slide 4, please. Slide 4. Here we are starting with a brief snapshot of SPARC, bit of a state of the union. As regular participants in this call, most of you are aware of our pivot from a delivery system innovation focus group to an innovator with reasonable appetite for early stage risk. We have made significant progress in that journey. Given where we have reached, I want to highlight 5 key drivers of SPARC's value upfront without dwelling too much into each one. We can go over this in detail if there are questions during the Q&A. First is the portfolio itself. 4 NCE clinical programs in areas of considerable unmet medical needs, Rhodobatnik in neurodegenerative diseases, and its companion program in late-line chronic myelogenous leukemia for patients failing on ponatinib. Rhodobatnik or SCD-044 in dermatology autoimmune conditions where S1P1 targeted therapies are evolving into an important class alternative to JAK inhibitors. And finally, our selective estrogen degrader SCO-120. And we are adding a very high potential platform built around a set of novel antibodies against what we call as tumor-associated antigen 1 to this mix. Including this one, on the preclinical side, we have now around 10 plus programs in various stages of the prosecution. So we believe our portfolio has enormous potential. Also better balance in terms of risk and novelty. SPARC is clearly going beyond our initial niches of validated biology and small molecule follow-ons. That transition is very real now. We have in the process become a balanced risk taker with differentiated assets pursuing validated biology on one hand, for example, BCR-ABL or SNTR1s, or completely novel mechanisms such as c-ABL inhibition in neurodegeneration or targeting new tumor-associated antigens with differentiated constructs. So our intent is simple: go after good science wherever possible in a modality-agnostic way when we have an opportunity to push the standards of care in difficult-to-treat diseases. So we are really proud to note that we have made important strides in that direction. Now, the third point here is the operating model itself. At its core, SPARC proposition is translational engine powered on actual brick-and-mortar lab infrastructure, not a virtual company, and high-quality human capital, which gave us substantially lower cost of failure. We have now 350 plus scientists with an overall head count touching 400 plus, 3 locations, 4 labs, and a self-sufficient process which can take a program all the way from bench to bedside. With significant validation in place, we believe our translation can be a real differentiator for SPARC. And on the other end of the chain, we maintain substantial flexibility. In the last few years, we have converted many of our late-stage assets from our legacy portfolio, Elysia with TriPoint, Cellpros with Sun, PDP-716 and SDN-037 now with Viziox, and Phenobarbital hopefully coming sometime in 2022. These transactions and potential royalties will certainly give us access to some non-dilutive cash. Not really enough to make our operations cash neutral, but this can contribute in a meaningful way and make a difference. These cash flows, with the recently raised preferential issue, allows us to keep most of the higher value programs in-house for a little bit more for the next set of inflection points, giving us a strategic flexibility to do something interesting hopefully in the future. And finally on this slide, the team. We have a truly global high-quality talent pool both in the middle to senior levels as well as at the entry level. Significant been there, done that experience at the senior ranks allows us to develop raw talent which is available in India. Add in a diversified and fairly accomplished advisory layer, we have the human capital to build a good quality operation. And that's a significant strength and means everything in this industry. So the broad point I want to leave you with from this slide is simple. For investors with appropriate risk appetite, looking for a bet in pharmaceutical innovation from India, SPARC offers a viable option with a high-value portfolio and increasingly validated operating model. Now, allow me to add a little more color to a couple of these points in slide 5, please. We have indicated in the past our operating model is getting built out fully with several important elements becoming more prominent now. Instead of talking through this whole slide, let me focus on a few important components I want to highlight. Accessing early-stage science is a do-or-die consideration for us. We've been building up our academic and small biotech outreach in a systematic manner in the last few years. We can now see the real impact of these relationships on our portfolio, both in our clinical portfolio as well as in the preclinical programs. particularly on the preclinical programs. We have structured relationships or ongoing collaborations with several Tier 1 academic systems globally, or their biotech spin-offs like WashU or University of Michigan or UCSF, increasingly Israeli universities on the university side, and companies like BioProjet or Biomodifying among academic spin-offs. These early-stage strategic partnerships are going to be a key element of our operating model. Our go-no-go decision-making has become substantially more stringent with external participation in the form of reviews with our scientific advisory board and also with external key opinion leaders. Idea is to kill unviable programs as early as possible and as completely as possible before it starts consuming significant discretionary spend during the costlier later stages of development. As we've spoken about many times in the past, our Indian footprint definitely offers significant cost arbitrage, but it goes beyond cost. There is certainly patient and data access advantages stemming from us being here, which can make our programs competitive. We are increasingly looking to leverage these data and patient advantages from India's additional opportunities to create value. And finally, on the commercial side, we now have several external commercial relationships, as I mentioned, TriPoint and Vizio and CMS with a bunch of our products in China. We are also reviewing additional external relationships for some of the other near-term licensing opportunities. These transactions offer important validations for our model, in addition to giving us some space to take the more valuable asset to later stages of milestones, which can give us better yield and realizations. So more broadly on this slide, we couldn't have been able to do the portfolio pivot which we managed without significant adjustments and capability development in the operating model, which we have done. And we are confident that SPARC represents a possibility for higher returns on innovation capital, as we can take significantly more shots on the goal for a given amount of invested capital. Now, a quick look on the portfolio strategy on the next slide, please. Here we go. As you know, we are focused on three important niches within our identified therapeutic areas: neurodegenerative diseases within neurology, treatment resistance in certain tumors in oncology, and some autoimmune disorders in immunology space. Why? Neurodegenerative diseases haven't seen much by way of innovation in a decade or two, making the standards of care there really stagnant. But we are seeing very attractive new science in this area, giving us exciting new testable hypotheses. We have to see this also with the progress in this field on the biomarker side of these diseases, particularly imaging markers, which give us a lot of anticipation about diseases like Parkinson's, Alzheimer's, and ALS. On oncology continues to be interesting for many reasons, in spite of high competitive intensity. Regulatory support for accelerated approvals and potential for premium pricing as two really important factors. And add in the mega basic science efforts going on in this space. Oncology is difficult to ignore for an early stage company trying to build a multi-therapeutic area portfolio like SPARC is. And finally, on immunology, several important opportunity areas. Localized immune suppression or modulation in diseases like Crohn's and ulcerative colitis, or effective oral options are missing in important dermatology autoimmune conditions where both the local options and more sophisticated biologics options are very strong, or for that matter, mechanistically synergistic combinations which can actually take efficacy to the next level are all opportunities in autoimmune spectrum worth chasing. And in terms of intent, we would like to have a multimodality toolkit, and we believe we now have a modular biologics platform which allows us enough flexibility to go after specific targets using multiple strategies. We call it molecular engineering for precision medicine, and we will talk a little bit about that later on when Nittan comes in. And we want to do that in short cycle innovation, fast hypothesis generation, testing and go, no go, and then repeating these cycles. So the key takeaway in this, the broader focus is on higher value, ripe for innovation areas with an intent to engineer solutions using a modular platform. Slide 7. So this is the story of our pivot, which I spoke about a little while back. I'm not planning to spend a lot of time here. Here are some highlights I want to leave you with. We're completing our legacy reformulation effort now. We have 3 NDAs planned for early part of 2022, as PDP-716, SDN-037, and Phenobarbital, and that completes our transition out of simple delivery system-based programs. I already touched on the clinical stage NCE, so let me not go there. We're planning an important preclinical platform play with antibodies targeting preferentially expressed tumor antigens. Here we have multiple possibilities to create ADCs, T-cell engagers, and other multi-specific entities. And these are very important additions to the portfolio, which can give us high impact, potentially first-in-class options going forward. And we will do a deeper dive on this part later on in the presentation. Before we transition to NCEs and biologics-based targeting, I guess it's important to talk about a few late-stage programs quickly, either in products in the market or going into a submission in short order. So very briefly, let me start with Elysia on slide 8. As you know, we have licensed Elysia to an early-stage U.S. neurology specialty called TriPoint Therapeutics earlier this year. And we've been working with TriPoint to launch Elysia in the U.S. We're very happy to note that TriPoint had a very good launch earlier this year, and they're scaling fast. TriPoint has around 40 reps now promoting the product across the U.S., and they plan to accelerate their rollout going into next year, and already have payer support from both Medicaid and also employer-provided systems. And we're very hopeful about the trajectory of this product going into 2022 and beyond. Now, on PDP-716 on slide 9. PDP-716, as you may know, is a once-a-day formulation of a widely used second-line glaucoma drug, brimonidine, which is indicated thrice a day. And we've had a successful phase 3 readout earlier this year. You may remember we also had a successful phase 2 study for this product. In these studies, we compared a 0.35% brimonidine once-a-day formulation with TID alpha-GAN 0.1% eye drops. And in the chart here, we have the data from the recently concluded phase 3 study. As you can see, the study measures intraocular pressure at 3 time points, 8 a.m., 10 a.m., 4 p.m., across 4 visits at baseline, weeks 2, 6, and 12. Across these time points, on multiple visits, the once-a-day brimonidine demonstrated comparable IOP reduction to alpha-GAN B thrice a day. The study also demonstrated comparability of safety profiles, 38.8% on OD and 32.3% to be precise on alpha-GAN P. These successful readouts set the stage for a submission early next year, calendar 2022, and the Viziox transaction, which we announced earlier this week. Now, let's also take a look at the other ophthalmic formulation, SDN-037, in the next slide. This 37, yeah. Now, SDN-037 is a twice-a-day formulation of an ophthalmic steroid used to treat the signs and symptoms of inflammation post-cataract surgery. Here in this phase 3 study, a statistically significant number of patients achieved an ACC 0 rating without having to take rescue medication compared to placebo. 68.3% on study drug versus 32.5% on placebo. SDN-037 formulation was very well tolerated and has an adverse event profile comparable to published difluprednate data. So as PDP-716, SDN-037 will also go into a submission in early 2022 calendar and forms part of the VCO transaction, which I will touch on next in the next slide. I'll start with the Ophthalmology Assets on the VCO transaction. As we have announced yesterday, we've licensed PDP-716 and SDN-037 to a US-based ophthalmic-focused startup called VCO. In addition to the regular licensing components like an upfront payment, milestones, and royalties in teens, this transaction also gets SPARC a 10% ownership of VCO. That allows us an opportunity to get an additional slice of their success beyond the regular royalty structure. We are super excited about our partnership with VCO and really looking forward to US NDA submission in early 2022 and subsequent market authorization. Another important transaction we recently announced is our licensing of a number of antibodies against a unique cancer target from a Israeli company called Biomodifying. As we've been talking about, we have a growing interest in biologics, and these antibodies will add to our ongoing efforts to leverage antibodies in oncology and also in inflammation, either as standalone therapies or as part of multi-specific combinations. We believe these programs will add significant value to SPARC's portfolio in the medium term. Now, let's move to the next slide to briefly discuss the ongoing impact of COVID-19 on our business. Slide 19. Slide 12, please. COVID has been a frustrating disruption for SPARC and also companies in our ecosystem. In many ways, the shutdowns and restrictions resulted in lost lab time, manufacturing and supply chain disruptions, leading to delays in the supply of investigational products, in some cases even other study materials, and also broadly clinical trial conduct across the board. The chart here on the right side of this slide summarizes the extent of disruption the clinical research ecosystem has taken in terms of delayed study starts, enrollment delays, even study cancellations and suspensions, so on and so forth. Even though we have done reasonably well on many fronts, we cannot claim to be an exception here. We've done very well with our lab operations. We've come back to almost full capacity early on in the pandemic without having to go back and shut down again during the Delta-induced second wave. We've done all right with drug supply with significant support from our CMC partners, including Sun. But we have been certainly impacted on the clinical trial execution, both in terms of site initiation, enrollment, and ongoing contact. The impact has been more in studies which are reliant on a single country for patient accrual or relatively rare populations. The Rhodobatnik CML is an example of such a setting, and we've seen some delays in that program. Our first task was to ensure continuity of patients who are already on the trial, both from an ethical and data preservation standpoint. We believe we've done well to ensure continuity of most patients in our trials through fair amount of virtualization and also through a mix of hybrid techniques. We've also de-risked our studies through significant geographic distribution, certain level of flexibility in our processes, and direct-to-patient outreach patient support services, and providing extensive supply chain and logistics support to our sites. We are hopeful going into 2022 to reclaim some of the lost ground through aggressive execution and direct engagement with sites and patients. Before we move on from this slide, let me say this. It's obviously an understatement to say COVID has been a major disruption. While we may not have mitigated our risks fully, we feel good about the proactive measures taken to deal with the pandemic and minimize the overall impact on our portfolio. I believe a lot of these operational process adjustments we've made during this phase will become standard practices in our system, with the potential to elevate the overall quality of our operations going forward. Now, before I transition, I want to briefly talk about our last fundraising round and plans for the future, and also briefly cover upcoming milestones. So let me go to the next slide, that's 13, please. Brief comments on the fundraising plan. Earlier this year, we have concluded a preferential issue of warrants to the tune of 1,112 crores, it's roughly $148 million. More than 50% or more, precisely around 600 crores, came from our promoter, and the rest from a group of genuinely marquee domestic investors and some foreign portfolio investors. I want to thank everyone on the call for their support and encouragement throughout this process. These warrants will be converted by the end of 2022. That will allow us cash to fund the development of programs like, say, Rhodobatnik, current trials in CML and PD, and SCO-120 proof of concept studies, and our ongoing discovery operations. This will also allow us to make necessary investments to build the capabilities necessary for newer modalities such as biology. But at the same time, our portfolio is expanding with multiple options for additional INDs in the medium term, in addition to additional indication options for some of the existing programs which need to be both preclinically and clinically prosecuted. We also need to plan the transition of current clinical programs to later stages of clinical development without taking major phase transition delays. With these considerations, we obtain shareholders' approval to raise up to 1,800 crores. We are in the process of evaluating our options for raising additional capital as we speak, and we look forward to your continued support as we move forward with this intent. Now, let's go to slide 14 and to take a look at SPARC's catalysts in the near future. On the legacy end here, we expect to see LHC and Cellpros growing. We look forward to working with Viziox to ensure NDA submission in the coming quarter for PDP-716 and SGN-037, setting the stage for NDA approvals and launch hopefully by early 2023. And we are working on a similar clock for Phenobarbital, and we hope to see a licensing event for that product hopefully in 2022. Now on to NCs on Rhodobatnik. PROSEEK and its companion trial in Lewy body dementia are both on track for data in 2023. Rhodobatnik's CML program is lagging our original schedule, as I mentioned earlier, and we hope to go into a submission in 2024 and trying hard to get back on some lost time as we see some normalcy post-COVID. On Vibostilimod, which is SCD-044, we hope to have clinical proof of concept for both psoriasis and atopic dermatitis by 2023. And SCO-120, our selective estrogen receptor degrader, is ramping up from its first-in-human SAD/MAD studies to patient trials as we speak. We expect to have early POC by the same time frame as 2023. In addition, we are also working towards moving at least 2 more programs to a U.S. IND. We will review one of those programs later in this presentation, and we hope to have at least 1 more IND in the same time frame. We will also stay in the hunt for additional assets from our external innovation pipeline. Slide number 15 has a nice summary of our programs and pipeline. I've touched upon most of this material in my comments already, so I don't plan to spend time on this slide. But this is a perfect segue to the next segment. That's Dr. Su Long Yao with updates on some clinical development programs. So I'll hand over the call now to Dr. Su, and he will talk you through these important programs, and I look forward to seeing you on the other end of the call. Thank you for the time again. Over to you, Su.
Thank you, Anil. My name is Su Yao, and I oversee clinical development. I also want to extend a warm welcome to all those who have supported us through the years, as well as newer folks who may be on the line now. We truly appreciate all your support and faith in what we do. So slide 17. Slide 17 presents some data that you've seen before in the past, but I've included here as a reminder for you. The diagram on the right is a kinome analysis that looks at potential targets of Rhodobatnik and shows that the activity of Rhodobatnik is specific against various related targets as opposed to many similar drugs where there is significant off-target activity. This allows us to hit the BCR-ABL target harder and potentially get more activity. The current plan is to study Rhodobatnik in the last line setting. That is, patients failing greater than or equal to 3 lines of therapy. We believe that this setting in highly resistant disease will best demonstrate the benefit of the highly specific nature of Rhodobatnik. As opposed to other drugs in the class, there have not been any QT effects, which can potentially lead to heart rhythm disturbances and death. Orphan drug designation has been received from the FDA and EMA, and as you know, orphan designation can extend the exclusivity of an approved drug beyond the conventional patent life. Finally, we've been fortunate in that the results with Rhodobatnik have been selected for either oral or poster presentation at several major meetings now. Slide 18. This slide is meant to give you a flavor of some of the results we've seen so far. This is a so-called swimmer's plot where the lines represent the duration of response. There are some caveats when you look at this type of data that you should keep in mind. Perhaps the most important to note is that the data are ordered according to when the patients enrolled in the study. So, for example, patients at the top of the figure enrolled most recently, whereas patients at the bottom were enrolled much earlier during the study. As a consequence, the patients at the bottom of the study have the potential to respond and stay on study the longest because they've had the potential to be treated the longest. Patients at the top of the figure, again, entered the study more recently, so that less time has passed, and of course, the potential to have a longer response is therefore not available to them. Nonetheless, you can see that there have been some marked responses, some of which have occurred at relatively low doses. For example, if you look at the bottom line, that subject has responded for over 4 years with a dose of 12 milligrams. The population summarized here is resistant or intolerant to at least 3 prior lines of therapy, so observing that kind of a median duration on study of nearly 21 months is notable under these circumstances. Slide 19. This slide summarizes the design of the ongoing study in CML and where we are. The study consists of 3 parts, a single ascending dose healthy volunteer component, a multiple ascending dose in patients with CML, and a pivotal efficacy part in patients who are refractory to greater than or equal to 3 treatments, one of which includes ponatinib. That's shown in the yellow there. As we previously discussed, there is general agreement between us and the FDA that this population in the pivotal study represents an important unmet medical need, and significant activity in this setting could provide the information needed for marketing authorization. We're most interested in the chronic phase group, since that's the largest group, of course, and there are a variety of sites recruiting in a variety of sites in a variety of countries, such as the U.S., Belgium, France, Italy, Spain, Romania, Hungary, Singapore, the UK, and Korea, and we're about a quarter of the way through accrual, with pivotal data anticipated to read out in 2024. Next slide, please. The next few slides, beginning with slide 20 here, provide some information on the use of Rhodobatnik for the treatment of Parkinson's disease. In particular, we believe that Rhodobatnik has most, if not all, of the properties needed to make a meaningful impact against the effect of c-Abl dysregulation in this disease. Rhodobatnik has sub-nanomolar potency and exceptionally limited off-target activity, giving it great potential to intensively limit c-Abl activity without causing unacceptable side effects. The ability of Rhodobatnik to penetrate brain with a brain plasma ratio of approximately 0.9 further augments Rhodobatnik's potential in the target tissue. The net effect is augmentation of autophagic flux and prevention of Parkinson-mediated mitochondrial control. All of this is technical scientific shop talk to basically tell you that Rhodobatnik is very specific for what needs to be done, has a unique ability to get to the place where the job needs to be done, and then gets the job done by removing the toxic alpha-synuclein that is the sine qua non hallmark of Parkinson's disease. Slide 21. This slide, so in the past we've shared cellular data on the effects of Rhodobatnik in Parkinson's disease. This slide gives you an example of the effects we've seen on behavior. As many of you know, Parkinson's disease is classically associated with tremors due to the death of neurons in the substantia nigra part of the brain. This slide depicts the results of a POL test in an experimental model of Parkinson's disease. TFF, or preformed fibrils, represent a method of inducing Parkinson's disease in mice. Mice that have Parkinson's disease due to TFF injection are unsteady and have difficulty in climbing down a pole, shown on the left here. It takes them longer to descend the pole. The graph on the right summarizes the results of treatment with Rhodobatnik in this model. On the y-axis is time in seconds required to descend the pole. Going across the bottom on the x-axis are various groups of mice. The first column in black there represents normal mice treated with placebo, and the second column in green shows the results from mice treated with Rhodobatnik only. The purpose of these two control groups is to establish a baseline for normal mice and to show that Rhodobatnik does not affect normal mice. The next three columns consist of mice which have had Parkinson's disease artificially induced by the injection of TFF. You can see that increasing doses of Rhodobatnik result in shorter descending times, so that by the time you get to the highest dose of Rhodobatnik, denoted by the yellow or orange column there, the time required for the mice to make it down the pole is very similar to the time required for normal mice to descend. In past presentations, we showed you that Rhodobatnik protects against neuronal loss in cellular studies. These data now show that the neuronal protection afforded by Rhodobatnik can have behavioral results, which may be ultimately clinically meaningful. Next slide, please. Slide 22 goes over the preclinical studies that we've done to support clinical studies and increase our confidence in Rhodobatnik and its safety. We've demonstrated the compound's safety and acute toxicity, single and multiple dose studies in mice, rats, and dogs by oral and intraperitoneal routes across a variety of exposure time frames. There was not any evidence of genotoxicity, meaning that the likelihood that chronic use could be associated with the development of cancer in humans is low. Reproductive toxicity studies have been completed across a variety of species, and as far as contact is concerned, there was only slight eye irritation and no skin irritation when the drug was applied to these surfaces. Finally, there was excellent cardiovascular safety in preclinical studies with minimal herd effects in vitro and no significant cardiovascular toxicity observed in monitored dogs. Slide 23. So one of the questions we've been running into recently concerns the results of a study that was performed with a similar c-Abl inhibitor drug called nilotinib, which is actually already approved and on the market for CML. In a study that was performed by the previous investigators, they evaluated 2 different doses of nilotinib against a placebo in patients with Parkinson's disease. The results of that study, as many of you may know, did not show much of a difference in outcomes between the 3 groups. In their conclusions, however, the investigators specifically stated that their results, quote, do not refute the hypothesis that c-Abl inhibition is potentially an important therapeutic target for Parkinson's disease modification interventions. Indeed, there are a number of novel molecules targeting the c-Abl pathway in development that have a better therapeutic profile, unquote. The data in this slide summarizes the properties of Rhodobatnik and its better therapeutic profile and why it's a better drug than nilotinib. To start off, we took a very systematic approach and first determined the concentrations needed in the cerebral spinal fluid in order for the drug to work in the mouse PFF model that I showed you earlier. We then performed a clinical study to make sure we knew the minimal dose that we needed to get that same concentration in the cerebral spinal fluid of humans. What we can tell you is that ultimately we have a compound that is 22-fold more potent than nilotinib, with which we are able to get 10-fold more drug into the CNS compared to nilotinib. The net effect is that potency with Rhodobatnik is around 200-fold greater than nilotinib. As I mentioned, this is all able to occur without cardiac rhythm disturbances or other toxicities that could limit Rhodobatnik administration. Part of this ability to intensely target c-Abl in the brain may relate to the fact that Rhodobatnik is not removed by drug pumps or efflux transporters, thus allowing it to cross the blood-brain barrier compared to other compounds. Slide 24. This slide summarizes the design of the ongoing clinical study in Parkinson's disease. There are 2 doses of Rhodobatnik that you can see there, and a placebo to which patients are randomized. Over the past year, we've added an open label extension, shown in gray at the bottom there, to obtain more information on the long-term effects of treatment and also to give patients an opportunity to continue to receive drug if they feel it's helping them. There are 84 sites involved in the study, and about 40% of the patients have been enrolled. Top-line data are expected in 2023. Next slide, please. The endpoints of the study that I just mentioned, the PERSEEK study, are summarized in slide 25. The primary endpoint is change in the Movement Disorders Society Uniform Parkinson's Disease Rating Scale, MDS-UPDRS, and secondary outcomes include the time to start of symptomatic medication, the EQ-5D score, this is a quality of life measure, the Clinician Global Impression of Severity score, as well as other pharmacokinetic and pharmacodynamic measures. We're also evaluating a few exploratory endpoints, such as change in dopamine activity in the brain, measured by DAT-SPECT scanning, along with skin biopsies, looking at the amount of toxic synuclein, and smartphone-based measures of motor performance and CSF, cerebral spinal fluid markers. Slide 26. This slide is here to remind you that we are also collaborating with a Georgetown investigator to determine if Rhodobatnik might be useful in Lewy body dementia. This disease has a lot of overlap with Parkinson's disease, and many suspect that the etiologies are actually similar. In this study, patients are randomized to either placebo or a low or high dose of Rhodobatnik. Treatment is for 12 weeks, and about a third of the patients have already been randomized. Endpoints include safety and tolerability, as well as a variety of biomarkers, and data readout is anticipated in 2023. Slide 27 moves on to vibozolamab, a sphingosine S1PR1 agonist that is being developed by us for a couple of inflammatory diseases. This is a background slide to give you some context for some of the subsequent slides. There are multiple drugs either approved or in late-phase studies in this class, including fingolimod, ozanimod, and etrasimod. Fingolimod was the pioneer in the class, but it's been associated with serious cardiac side effects, possibly due to its effects on the S1PR3 receptor in the family. Currently, vipazolimod is in early phase 2 studies in atopic dermatitis and psoriasis, with readouts expected in 2023. I'll provide you a little more detail about this in the next few slides. Next slide, please. Slide 28 provides a little more information on what we're seeing with vipazolimod in humans. We initially studied vipazolimod in a 3-part study consisting of a single ascending dose part, followed by a food effect part, followed by a multiple ascending dose component. In general, vipazolimod has performed the way that we anticipated based on the preclinical data. In the single ascending dose portion of the study, 6 dose levels were studied in males and 1 in females. There was a 55% reduction in lymphocyte count following a 1 milligram single dose. You can see this in the figure on the right. This magnitude of effect is expected to be associated with efficacy, as has been observed in other drugs in the class. There's no food effect observed in the food effect portion of the study. 4 dose levels were studied in the multiple ascending dose portion of the study. Approximately 60% reductions in lymphocyte counts were observed following 1 milligram doses, and representative pharmacokinetics are shown at the bottom right-hand side there, with a half-life just shy of 3 days. Slide 29. This slide summarizes the design of the ongoing phase 2 study in psoriasis. In this study, 240 patients are randomized among 3 doses of vipazolimod and placebo and treated for 16 weeks to obtain results on PASI 75, the primary endpoint of the study. This is shown in the pink or magenta color. There are a series of subsequent re-randomizations shown in yellow to provide insight into things like the durability of response on therapy and the durability of response following cessation of therapy. The study has just started with approximately 10% of the subjects randomized. Next slide, please. Slide 30 goes over the design of the atopic dermatitis study. This is actually quite similar to the psoriasis study. This study also consists of 240 patients, approximately, randomized to 3 different doses of vipazolimod or placebo. The endpoint is slightly different. It's the EASI-75 instead of the PASI 75, but it also occurs at week 16, and the study also recently started. Slide 31. This slide provides some background on our oral selective estrogen receptor degrader. As you may know, this is a substantial market. Approximately 200,000 women are diagnosed with estrogen receptor positive disease, and as you can see on the right there, survival can be poor in advanced disease. These women, along with the 20 to 30% of them who acquire mutations, would be eligible for treatment with SCO-120. Oral SCO-120 is designed to be a step above intramuscular fulvestrant, which is both uncomfortable to receive, as well as very poorly active or even inactive against estrogen receptor mutations at practical doses. We're in the midst of transitioning from healthy volunteer studies to patients at this point. In general, I can say that SCO-120 has been very well tolerated and has been no really notable AEs. The multiple dose study in patients will be starting soon, and patent expiry is expected to be around 2040. Next slide, please. Slide 32 here summarizes some preclinical efficacy data with results in mice with brain metastases. On the y-axis there, you have the proportion of mice with brain metastasis who survive. On the x-axis is time in days. Treatment is started on day 7 and continued for 8 weeks. There are a lot of curves there, so some of which can be difficult to distinguish, but I'm going to ask you to focus on the x-axis and the curves that end around 98, 84, and 77 days. Those are the light blue, or turquoise, orange, and red curves that end around those days. Those groups of mice, you can see, have the longest survival, and those groups are the groups who have been treated with the high, middle, and low doses of SCO-120. There's another curve in gray as well as another curve which is buried in there around day 35 with the control groups. And that gray curve is fulvestrant and the radius compound. And you can see that survival with fulvestrant is not much better than the control groups. In fact, this is consistent with what we know clinically, namely that fulvestrant does not penetrate well into the CNS or central nervous system, and it's not a good treatment for patients with brain metastases. In short, these results suggest that SCO-120 may have activity in patients with brain metastases as well as tumors elsewhere in the body. The next slide, slide 33, goes over our current clinical plan for SCO-120 in patients with breast cancer. We've completed dose escalation now in volunteers and are at or near exposures that we expect to be efficacious. We will confirm the tolerability of these doses in patients and see if we can escalate a bit more in part 1 of the study that's soon to start. Altogether, this will probably involve around 15 patients. The main focus will be to confirm the safety and pharmacokinetics we saw in volunteers in patients. Part 2 of the study will focus on obtaining preliminary efficacy information and pharmacodynamic confirmation of the dose to be used for later phase studies. There'll be around 15 patients involved in this part of the study, and the study will start shortly and provide results in 2023. The next slide, slide 34, goes over the last drug that I'll touch on in my portion of the presentation. Phenobarbital, as many of you may know, is a well-known existing drug for the treatment of seizures, and previous studies have shown that its activity can be superior to that of existing anti-epileptic drugs such as levetiracetam. However, its current formulation has some limitations, especially when it's used for the treatment of seizures in neonates. As noted in the second bullet here, the current formulation contains benzyl alcohol as a preservative. Unfortunately, benzyl alcohol is toxic to neonates and can cause a gasping syndrome, which can ultimately be fatal. There have been many guidances and advisories which warn against the administration of drugs containing benzyl alcohol in neonates. What we've done here is remove the benzyl alcohol so that this will no longer be an issue in the treatment of neonatal seizures. SPARC's formulation has been granted orphan drug designation by the FDA, and we're anticipating filing for approval sometime in 2022. I hope I've been able to get you up to speed on some of the more important clinical stage programs in our pipeline. And at this point, I'd like to introduce my colleague, Dr. Nittan Baumler, who will tell you about something perhaps even more exciting in the preclinical space. Nittan?
Thank you very much, Sue, and good evening to you all. I'm Nittan Baumler, and I look after the biologics R&D at SPARC. In the next 10 minutes or so, I would like to discuss our biologics space, which is a new dimension for our R&D activities within SPARC. We had noted in our presentation to this audience a year ago that we have been exploring development of a new biologics technology platform with the potential to yield multiple molecularly engineered and innovative precision medicines. This modular platform technology has evolved from the antibody engineering advances made over the last 20 years or so and leverages the applications of single-chain FV as a unit for antigen recognition and binding, as shown on slide number 36. Using such modularly assembled SCFE immunofusions can be used as a substitute for a classical tetrameric IgG and can do almost everything that classical IgG antibodies can. This platform allows us to make use of any antigen specificity of choice and thereby create mono, bi, tri, or even tetra-specific immunofusions with the power of effecting functions of as many as 4 distinct antibodies. So we can incorporate in principle power of 4 different antibodies into one molecule. We have been able to create such multifunctional immunofusions with anti-tumor and anti-angiogenesis effects for use in cancer therapy and anti-inflammatory immunofusions for the use in inflammatory diseases. And these are currently being explored for therapeutic impact. What I would like to share with you here at this point is a glimpse of our very first biologic asset being developed as an antibody drug conjugate, or ADC, targeted to as yet undisclosed tumor-associated antigen 1, or TAA1. The concept of ADC is pretty straightforward. ADCs represent antibody-targeted delivery of chemotherapy, in which a potent cytotoxic drug is covalently linked to an antibody with a specificity for a tumor antigen that is often overexpressed on the surface of tumor cells, and via which the cytotoxic agent can be preferentially delivered to the targeted tumor cells. Thus, delivered cytotoxic agent then kills the tumor cells but spares the rest of the body from the toxicities of the drug. ADCs are rapidly becoming a staple in cancer therapy, with the regulatory approval of at least 11 distinct ADCs in the US and Europe over the last 20 years or so. In our very first ADC asset, we have made use of clinically and commercially validated drug linker combination to create our own ADC. The target of our ADC is an antigen, here referred to as TAA1 as I mentioned earlier, which is overexpressed on the cell surface of a wide variety of human carcinomas, including those that have relatively fewer treatment options. Additionally, when antibodies bind to TAA1, they are rapidly internalized. Hence, TAA1 can be an ideal tumor antigen for targeted delivery of potent cytotoxic payloads or other response modifiers. Here, I would like to share with you a glimpse of in vitro as well as in vivo activities of the TAA targeted ADC. Next slide, please. Slide number 37. Here we show the in vitro growth inhibitory effect of the ADC against pancreatic cancer cell line that overexpresses TAA1. The left side of the graph shows that TAA1 targeted ADC in a concentration-dependent manner inhibits the growth of this cancer cell. In the graph on the right side is the effect of a similar ADC made using a CD20 specific isotype matched antibody that does to carcinoma cells. This non-binder ADC is able to exert cytotoxic effect only at the highest concentration tested and is about 50 to 100-fold weaker than the TAA1 specifically targeted ADC. Similarly, although not shown here, TAA1 specific ADC is unable to mediate preferential cytotoxic effect against carcinomas that lack the expression of TAA1, indicating that the anti-tumor effect of TAA1 specific ADC is specific and focused for TAA1 expressing tumor cells. We have further evaluated TAA targeted ADC in multiple human tumor carcinoma xenografts established in immunodeficient mice. Next slide, slide number 38. Here we have evaluated low, intermediate, or high dose of anti-TAA1 ADC in the pancreatic carcinoma xenograft that expresses high levels of TAA1. In addition, we have also evaluated the same ADC in an ovarian carcinoma xenograft that expresses relatively low levels of side of the graph shows the impact or the anti-tumor efficacy of this ADC against pancreatic high TAA1 expressing carcinoma. On the right side, you have the same ADC being evaluated against low TAA1 expressing carcinoma, ovarian carcinoma. In this evaluation, the putative therapeutic therapeutics were administered in tumor-bearing animals once a week for 3 weeks, after which the growth of the tumor xenografts was monitored. The anti-TAA1 ADC, in a dose-dependent manner, strongly inhibits the growth of both high and low TAA1 expressing carcinoma xenografts. In contrast, neither the unconjugated cytotoxic drug used at the same concentration as that of the highest dose of the ADC, nor the non-binding anti-CD20 ADC at the highest dose can exert meaningful growth suppressive effect against these carcinoma xenografts. We are presently evaluating the impact of TAA1 targeted ADC against human carcinoma xenografts that express varying levels of TAA1 to further establish the TAA1 specificity of this ADC. Although not shown here, anti-TAA1 ADC can suppress the growth of TAA1 expressing carcinomas irrespective of their size. So both large and small volume xenografts are subjected to therapeutic impact and have shown to be inhibited in similar evaluation. We are excited about these clinical results and hope to advance these ADC through preclinical development with an eye on the IND submission targeted for 2023. Next slide, please, slide number 39. We are exploring a number of distinct ADC opportunities that can benefit from the experience of developing anti-TAA1 ADC. Our TAA1 focused efforts are not limited to just the ADC platform. Using TAA1 targeting as an anchor, we are also exploring biologic strategies to engage the immune system much more effectively for effective cancer treatment. In this regard, we are also exploring T-cell engaging strategy using our bispecific immunofusions in which one specificity can be targeted to the T-cells and the other being TAA1. Using such a T-cell engager, tumor cells and T-cells can be brought together, resulting in the activation of T-cells and their subsequent attack on tumor cells. In another version of the same strategy, immuno-enhancing cytokines or co-stimulatory ligands can be used as part of the bispecific or trispecific immunofusion assembly. However, these remain quite exploratory at this point for further elaboration. What we would like to emphasize here is that this immunofusion platform has the potential to represent multiple first-in-class biologic products for our near-term pipeline. Next slide, number 40, please. While I have discussed so far the biologics aspect of our R&D, there are a number of small molecule or NCE opportunities that we are pursuing. And there are two such opportunities that I'm going to mention in this presentation. The first is a synthetic targeted cytotoxic agent, in which the cytotoxic payload is linked to a small molecule ligand targeted to a tumor-associated antigen. So in similar vein as ADCs, except in this case, instead of using antibody, we are using a small molecule ligand as a delivery vehicle. And an application of this is intended for a metastatic prostate carcinoma. The second small molecule opportunity that I would like to refer to here is an immunomodulatory agent that we would like to develop for the treatment of alopecia areata and possibly other immunoinflammatory disease indications as well. You may learn about these opportunities in our subsequent presentations in the years to come. I will stop here and would like to invite Chetan Rajpara, SPARC CFO, to provide financial update. Chetan?
Yeah, thank you.
This is the operator, so we are not able to hear you.
Can you hear me now?
Yes, sir.
Yeah. Good evening, everyone. This is Chetan Rajpara. I plan to go over SPARC's financial and cash position at a high level. Slide number 42, please. During FY21, total income was rupees 258 crores, while total expenses were rupees 410 crores, resulting into a net loss of rupees 151 crores. I would like to mention that FY21 income includes an upfront payment of $20 million from STD 044 licensing deal, which is a non-recurring item. Let me update you on our financial results for the first half of FY22. During the first half year FY22, total income was at rupees 56 crores, while total expenses were at rupees 173 crores, resulting into a net loss of rupees 117 crores. Next slide, please. Slide number 43. As Anil mentioned earlier, the company has raised rupees 1,112 crores in July 21 by way of a preferential issue of warrants in order to meet expenses over next 24 to 30 months. Of this, rupees 600 crores were contributed by promoters and remaining rupees 512 crore were from 30 external investors, including 8 FPIs. The company has received rupees 278 crores upfront, being 25% payable by investors on application. The balance rupees 834 crore is to be received upon the conversion of warrants by investors within 18 months from the date of allotment. Cash on hand as on 30th November 2021 was rupees 12 crores. The company also has a line of credit for rupees 250 crores from the parent company, in addition to the bank facilities for rupees 218 crores. Of these facilities, we have used 100 crores as on 30th November 2021. The company has obtained shareholders' approval for raising an additional sum up to rupees 1,800 crores by way of issuance of fresh equity or debt. The company is in process of licensing certain late-stage clinical assets in order to generate additional liquidity. For FY22, approximately 40% of our expenses are budgeted for the clinical cost. However, we are aggressively managing our cost and working to control our non-clinical expenses tightly. The company has taken all possible measures to limit the impact of COVID in order to ensure business continuity with minimal disruption. The company will further evaluate and actively respond to minimize its impact on the financial performance. That's all from me today on the financial update. A big thanks to all for joining the call. I will now hand over the call to Jaideep for facilitating the Q&A.
Thank you, Chetan. We will now open the call for question and answer session.
Thank you very much. We will now begin the question and answer session. Anyone who wishes to ask a question may press star and 1 on their touchtone telephone. If you wish to remove yourself from the question queue, you may press star and 2. Participants are requested to use handsets while asking a question. Ladies and gentlemen, we will wait for a moment while the question queue assembles. Reminder to the participants, anyone who wishes to ask a question may press star and 1 at this time. The first question is from the line of Ketan Gandhi from Gandhi Securities. Please go ahead.
Yeah, sir. Any guidance on PDP 716 and SDN 037 for peak sale potentials? I believe it has reduced substantially since the last presentation.
Hi, Ketan. This is Anil. We have been made forecast projection for PDP 716 or SDN 037. So even in the last presentation, so we will even in this edition, we are not making a forecast for the program, and we will we are working towards a NDA submission sometime in early part of 2022, and subsequently a launch in 2023. And as we work with the partner, we will try to give market a guidance maybe towards the launch.
All right. And any guidance for Phenobarbital?
Phenobarbital also is early for us to make a guidance. We need to first go through the process of licensing the program out, which we are hopeful we can complete in 2022, and then we will work with the partner to give a guidance to the market.
All right, sir. Thank you so much.
Thank you.
Thank you. Participants, to ask a question, you may press star and 1. A reminder to the participants, anyone who wishes to ask a question may press star and 1. The next question is from the line of Manish Jain from Gormal One. Please go ahead.
Yeah, hi. First of all, congratulations to the entire team on such great progress despite COVID-led massive challenges. So my first question was on Rhodobatnik, that what are the scenarios under which we can get maybe an accelerated approval ahead of the timelines that we have shared in the presentation, where the kind of clinical results, if they are far ahead of what we are budgeting, if you can give some scenarios, that will be helpful.
Thanks, Manish, for your kind words. On the question specifically on Rhodobatnik in terms of the possibility for an accelerated approval, that I will be stepping into a speculative realm if I say, specific scenarios. I mean, we are, as you can see, we're doing a fairly well-powered clinical study. And the idea is to create a reproducible outcome from this PROCEED program, and also provide definitive proof of concept for the seeable hypothesis. And if we have what we hope to have, which is a positive outcome from this trial, at that point, we need to initiate a conversation with the agency. The agency has been fairly encouraging of new neuroprotective interventions, as you have seen in the recent Biogen controversial, but the Biogen approval, which also is leading to additional submissions in the editor pathway in Alzheimer's. So we're going to have a conversation in terms of where we can go with the results. And there are several possibilities, and classically, if you look at the pathway for approvals in these settings, it would require 2 additional phase 3 studies, and the rest of it is a matter of negotiation in terms of whether there is a possibility of accelerated or a conditional approval, and then following it up with additional studies, or getting an approval with 1 additional study. They are all in the speculative domain, and I, at this point, need to stay away from that.
Great. No, that answers the question. And Chetan, just one clarification, that you've mentioned the cash position as of 30th of November 21. Has these been after the upfront payment receipt of Viziox and the upfront payment after paying to Biomodifying LLC? Has both of these been accounted for?
Manish, both these payments are yet to be processed. So this position is before making this payments.
Perfect. Thanks. I had few more questions. I'll join back the queue.
Thank you, Manish.
Thank you. The next question is from the line of Anubhav Agarwal from Credit Suisse. Please go ahead.
Yeah, thank you. Good evening to all. Couple of questions. First is on Vibozilimod, where we have partner Sun Pharma. So just a clarity there on phase 2 trials, is Sun Pharma going to fund the trial, or you guys will fund it and then it will get reimbursed? Can you clarify that?
No, this program is the clinical program for Vibozilimod post phase 1 is funded by our licensing partner at Sun Pharmaceuticals.
Okay, so even phase 2, which are going to start for all the immunology sites, will be funded by Sun Pharma.
Okay, it's already, these studies are on, so it is not going to start. They're currently recruiting at the moment.
So if I see a pipeline, majority of spend for you for SPARC will largely go towards Rhodobatnik, right? Would that be
We have, 3 studies in Rhodobatnik, that is Rhodobatnik Parkinson's trial, which is a fairly large global program. We have a smaller phase 2 program, which is a single center study with Georgetown in a form of Parkinsonism called Lewy body dementia. And then we have multiple studies on a multi-part study for registration of Rhodobatnik in chronic myelogenous leukemia. And then we're also ramping up significantly on SCO-120, which is our estrogen receptor degrader study, which is moving now from a healthy human volunteer SAD-MAD leg to actual patients, which is going to consume resources.